My name is Olatunde Michael. I am here once again this afternoon to talk to you as a potential bioinformatician in most of these biotech industries. Don't forget that in the introductory part of bioinformatics, I have actually introduced you to uh, the basic, the basis of bioinformatics. And just like what we know from the name Bioinformatics, that is an application of bioinformatics. So what we are here today to do is to prepare ourselves by reviewing most of the earlier videos on bioinformatics. So we'll review most of our past bioinformatics videos. We'll be able to review them and make them to be able to prepare for any professional examination that has to do with dealing with multi-choice questions. In the in the quiz that will be posted to the comment section later on, it is going to be made up. It is going to be divided into two parts. The first section is going to deal with introduction to bioinformatics. That is, you are going to have 30 questions, between 30 and 50 questions, that is going to test your knowledge in introduction to bioinformatics. It's going to test your knowledge in what bioinformatics is. Don't forget that it is the study of biological information, and that the primary application of bioinformatics is to use it in drug discovery. We also use it in vaccine design. We can also use it in other areas of life. And the data format that we normally make use of in bioinformatics is called FASTA. So you have to be very, very familiar with FASTA format, because most of this information will be retrieved from the database in FASTA format. And there's a format for that. FASTA format, we always start with greater than, and you have every other thing that's going to follow. So there's a way you can convert the nucleotide bases. You can also convert nucleotide bases manually. You can also convert them into FASTA formats. Then we also make use of basic local alignment sequence tool, that is blasting. Blasting is a tool that we use to check for the homology, how similar certain two sequences could be, or how similar, how dissimilarly they are even different. So we normally employed blasts as a kind of tools to interpret all these biological data. Then and the bioinformatics, in bioinformatics, the database we normally use, there are different types of a database. We have a gene bank, we have UniProt, we have a protein database. Depending on the sequence, the type of sequence we have at our disposal. So if we are with the DNA sequences, we actually know where to go to. We normally make use of NCBI for that. And if we are with protein database, or that is amino acid, we know that we actually go to Uniprot, protein database, Swiss model, and so on and so forth. And the language the language that we normally use in the bioinformatics is they normally use Java. Some you use Java, they use Python is the language in bioinformatics. Then apart from that, we have actually I also exposed you to some of the sequence alignment tools that you can use to compare large DNA. You use sequence alignment to compare large DNA sequences. Apart from that, you should also know what is called a GO, that is gene ontology. What gene ontology alignment tools does is that it annotates protein functions. So that is the sequence you use, and it also has other tools that you use for drawing the phylogenetic tree. So you can use different types of tools in understanding that. And if you also want to predict the structure of protein, especially the structure of protein, don't forget that proteins, when they exist in amino acid, they are said to be at the primary level. And with this primary level, you can also go to higher level by trying to understand the folding of this amino acids, to be able to know their folding. If you want to know their function, the function they will always be. That means you need to know the secondary structure. And if you want to know the secondary structure, there are different types of bioinformatics tools that you can use for that. This includes Pirate2. You can use Pirate2, you can use PyMol, because so many of them. So to be able to use it for your structural design of a bioinformatics. And there are higher level, like if you want to know the three-dimensional structure, that is when you want to know what is going to be the bond, the interaction, that is ligand and chemical bonding that has to do with docking. And that is exactly what we normally believe in and what we normally use in docking. So we normally use that. So just like I told you that we are going to have two different tasks that will be given in this lecture this evening. The task one is already on the screen. So you have to tell us what do you know by what are biological databases, what do you know about them, and you should be able to list any 5 databases that you know, and you should be able to also state their functions as well. So the task should be submitted and posted to the comment section of the under this lecture thereafter, and that will serve as a kind of a feedback into what we have been doing so far. So the task 2 will also be given during the course of this lecture as well. So don't forget that I said the MCQ questions, which is going to be made up of 60, at least minimum of 60 questions. 30 will come from to test our knowledge in introduction to bioinformatics. You need to familiarize yourself with what is bioinformatics, what are the disciplines that will come together to make use of bioinformatics, what are the applications of bioinformatics, how can you apply bioinformatics in tech, that is in the industries, and in the modern day of bio. So don't forget that bioinformatics is it is the present and it is the future of biology, because it has a lot of relevance. It has a lot of relevance. Its relevance in drug design is cannot be overemphasized. In personalized medicine, it is very relevant, and also in cancer, that is ontology. In cancer therapy, immunotherapy, all this one They are also very, very important. So, and I want you to know, you should be able to know if they ask us to, if they ask you to write short notes on the following: FASTA, BLAST, GenBank, UniProt, PDB, Python, Cluster W, GenMark, Gene Ontology, that is a GO. You should be able to say something about PDB. I've said that. Then Galaxy. Galaxy is a model that they use to refine the secondary structure of protein. Then you should also know something about what is called multiple sequence alignment tools. So multiple sequence alignment tool, that is a MSC, is what we use to establish the evolutionary relationship in a given situation. Okay. Then apart from that, you should be able to know that the programming language in bioinformatics is Java. Python, as well as C++. All these are part of the languages that they normally use. Then don't forget that in trying to determine gene expression in any organism, so the technique that you use is also called a microarray. And just like you know that the term genome is often referred to the set of entire genes, the totality of entire genes in any organism is known and called the genome, and the totality of the protein in any system is the study of proteomics. The same thing with metabolomics as well as transcriptomics. Okay, you need to be familiar with this. And just like I said, that one of the computational application of bioinformatics is that the computational biology will actually help us to be able to use algorithm for our biological data. So by the time you understand some part of computational biology, that one will help you to be able to set up or draw an algorithm that is going to help you to be able to interpret, utilize, and analyze your data, biological data. Anywhere you come across data in this course, is nothing but it's referring to biological data. And I told you that you should be familiar with what is called protein folding. Protein folding. The more folded a protein is, the more functional that protein is going to be. So, and the amount of folding in any specific protein will depend on how much it is going to be functional. So don't forget that I've also said this in microbial genetics when we are talking about translation, that there are some post-translational mechanism or post-translational activities that will most of this protein will undergo in the process of translating the messenger RNA into protein. So the more folded the protein is, the more functional it is going to be. And that is the more it is folded, that is exactly what is going to lead to the graduation of protein into, and their calibration into primary, secondary, tertiary, and the newest one, which is at the 3D structure. So when you talk of primary structure of a protein, that means you are looking at the sequence, the amino acid sequences in that kind of a protein. And by the time you are talking of a secondary, that means you are already talking of whether it's going to be alpha folded, it's going to be beta folded, or it's going to be anti-clockwise or clockwise, or it is going to be the mixture of that two. And that is the level you are going to make use of different. So there are, depending on the level of protein that you are actually interested in, so that will now justify or help you to be able to know the type of bioinformatics tools that you are going to use in interpreting it. So for example, now in the area of predicting the secondary structure by the use of a pyrrole, by the use of a pymol, you know what and what PSI pred, you know how it is going to be appearing in terms of their bonding level with hydrogen sulfide and all the rest. So the amount of folding or the folding of any protein is very very useful for us in structural biology, in understanding the structure. Well, if you understand the structure, that will definitely help you to be able to understand what the function or what the function it is going to be in a certain organism. So then you also know to have come across the word immunoinformatics. Immunoinformatics is nothing that application of bioinformatics to be able to understand immune system, and that means if you don't have very good knowledge of immunology, you might not be able to actually understand what immunoinformatics is all about. It is very very applicable, and that was what we have actually dedicated some of our past few videos. I'll be able to dedicate it on understanding what immunoinformatics can do. Can we simulate the immune system for vaccine design, for drug design, and as well for predicting whether there's going to be some cells are actually being vulnerable to cancer, and while some are actually not vulnerable. So that is going to help us in understanding what is called biomarkers that can actually help us to be able to diagnose all this very very well. So I told you that the primary goal of immunoinformatics is to be able to predict the immune responses. So that is the first goal of that. Then apart from that, the goal at least apart from that, you can also use it to analyze the antigenic properties of any given protein. If you are given any protein and you want to understand whether it is going to be useful immunologically, so we use what is called immunoinformatics to be able to analyze that antigenic properties of that protein. Then, as I told you, we also use immunoinformatics in vaccine design, and to be able to understand the mechanism by which immune system will actually work. And some of the methods that you use immunoinformatics include: we make use of immune epitope databases. You have to be familiar with that. IEDB, immune epitope databases. It's very very important. By the time we actually start vaccine design and docking, we are going to see some of this. And epitopes. Okay, many of us might be coming across the word epitope for the first time. Epitopes are nothing but the certain segments or the certain portion of an antigen is actually being regarded as a epitope. So when you see any antigen, it's not going to be the whole part of that antigen that is going to actually trigger an immune response. It will just be certain portion. It could just be maybe few amino acid sequences of amino acid that will be the epitope in that antigen, and it is by the use of immunoinformatics that we can use to be able to predict that. Then we also come across the tools that we use in predicting the MHC binding properties of peptides. We also come across that as well. So after that, you also need to know. Then what do we actually refer if we say immunogenicity? Immunogenicity is the capability or the ability of any antigen or any substance to be able to trigger or provoke an immunological response. So at this level, I'm very sure that you're supposed to have known exactly what we call in silico In silico is maybe is an arrangement. If you say in silico vaccine design, in silico vaccine design now is nothing but the use of a computer to be able to design a vaccine. So that is very, very important. Then apart from that, um, there are other epitopes as well. There are other databases that you use in predicting the epitopes. So the more you are able to predict the epitope, the more likely for you to be able to design a very effective vaccine candidate for that antigen. So you need to know that. And apart from that, you at this level you should know that there are two arms of immune system. There are two arms. We have the innate immune system and we have the adaptive immune system. And don't forget that immune system actually work in form of a kind of intricate network of cells coming together to collaborate in order to be able to protect the host from the effects of antigen or pathogen, so to say. So it is a part of the immunological or immunoinformatics that we can use also to be able to understand this one as well. And just like I said, fascination is just a principle that is being followed whenever we want to design as well. So then apart from that, you need to also know you need to know that in immunoinformatics has actually helped us to understand a lot of things. That's helped us to understand a lot of things about how what is a personalized medicine. What do we mean if we say personalized medicine, which is an individualistic way of responding or treating or diagnosing any kind of infection. So part of what we normally use, the role B cells played in immunoinformatics, primarily what B cell do in immune response is nothing but production of antibodies. And don't forget that we have five classes of antibodies, which I mentioned yesterday. We have immunoglobulin G, we have A, we have M, we have G, we have E as well, depending on the level of involvement of that kind of antigen. So that is very, very important. Then also in predicting or design of vaccine, you need to familiarize yourself with some other databases like NetMHC, Swiss model. You need to also familiarize yourself with what is called adjuvants. What are adjuvants? Then is going to be effective or is not going to be effective. So you need to test this as well. So that is very, very important. So then apart from that, you need to familiarize yourself by knowing some other things in immunoinformatics. You need to familiarize yourself by knowing cancer immunotherapy. Then apart from that, you also need to know most of what we at a point in time, if always we also mention the function of immunotherapy or immunoinformatics in cancer therapy, that is in production of cancer drugs. Yeah, in production of drugs that can be used to cure cancer. And don't forget that anything that is going to trigger an immune response, we ought to know the mechanism of the action of that immune response. We need to know that. So it is very, very important. Then, not only that, which of the, for example, if there is a question of immunoinformatics in cancer design. So you can make use of this in of it, designing cancer vaccine. Not only that, you can also use immunoinformatics in predicting immune. You can also use it to target. You can use it to target tumor specific and so on and so forth. So you need to know this. Then some tools, like if you are to identify those ones that cannot be used in immunoinformatics, the only one that you cannot use here is you can use blasting. You need to do blasting. That is when you want to do sequence alignment. That is to the first thing is that when you are given set of antigen, set of sequences, the first thing you need to do is that you go and do what is called homology testing. If you are given sequences, you retrieve those sequences from the database. You go and test whether that sequence will have the ability to interact with the gene of the host, that is of man. So if it has a kind of interaction, and what you are going to make use of is you make use of blast. That is, you blast. Immediately after blasting, you can now continue. So you make use of blasting. We also make use of net MHC. I've told you. IDB, we make use of that. So you need to know this. Then apart from that, which process is used to improve the immune response against a pathogen in a vaccine design? And that is nothing but epitope mapping. So don't forget that I told you what epitopes are. And I told you that epitopes are nothing but a segment of an antigen that can trigger an immunological response. So the very active point of any antigen, that is the one that the pathogen will always make use of in order to be able to be pathogenic, that segment is called the epitope, and that is the segment at which the antibody will be able to bind to. So that point, the exact point which is antigenic in any antigen, is called the epitope. So, and the more you are able to do the mapping, that is, you are able to get some of the higher the epitope, the better the vaccine that you are going to design by the use of that. So if you have just one binding site, okay, so epitope are the binding sites for antigen. Is better, yeah, I think it's better to say that is the binding site for an antibody or for immunological for any immunological item. So that is the antigen binding site, that is called the epitope. So the more the epitope in any given vaccine, the better that vaccine is going to be. So, and that is exactly what, and one of the reasons why you need to do what is called epitope mapping. Epitope mapping is very, very important that you do it in immunoinformatics. Then not only that, you also need to do a lot of simulation of how antigen is going to bind, at what point is going to bind to. That is the T cell receptors site, as well as the vaccine candidate that you have actually designed. So it is very, very important that you also go into doing that as well. So, and that is exactly, you also need to do what is called docking, so which has to do with the best suited sites. It's just like a lock and key. So the key hole is going to be on the surface of the antigen, and the real key that is going to enter is going to be the ligand, which are primarily available, and which they normally use as a binding tool. So there are going to be in immunoinformatics to be able to do or predict what is going to be the antimicrobial resistance. So that is very, very important. So don't forget again that is going to be kept in the comment section. So you just attempt them, and you are also going to, you have to submit as well. So you see this in the comment section. So don't forget to like this video and make comments. So thank you, and God bless you.